Hey Team Bio, welcome to your screencast on protein synthesis and secretion. In this screencast, we're going to be talking about how proteins are made. So the process of taking DNA, oops, this is way too big. The process of taking DNA, oh, that's too small. Gotta find the Goldilocks size of a pencil. Okay, DNA and reading it to make RNA and using RNA to make protein, which proteins are like the action organic molecules in your cells. They lead to your traits and behaviors. And then, so this is all part of protein synthesis, DNA to RNA to protein. This bit. And then secretion is moving these proteins through the endomembrane system. And the endomembrane system are the true organelles. Of the cell. So we're using protein synthesis and secretion to tie together what's the central dogma of biology, how DNA encodes our traits and behaviors. And then we're tying that together with the endomembrane system or the organelles that we're studying in class. So I'm going to give you this screencast, and then you are going to take what you learned in the screencast and apply it to a new set of diagrams in class and create your own screencast talking about how catalase, the protein that we studied in the fall term, it decomposes H2O2 into O2 and H2O. This is an unbalanced chemical equation. So it's the enzyme that catalyzes this reaction. You're going to use this screencast to explain how catalase is produced in the cell and then how it ends up outside the cell because it is a protein that is secreted. Okay, so this whole process begins in the nucleus with the DNA. DNA is a code. It's comprised of nucleotides with the letters A, C, T, and G, and the order of those A's, C's, T, and G's is important. Just like this sentence, the cat ate dog means something. If we rearrange this to say dog ate cat, it means something different. The order of the nucleotides in the DNA, the sequence is important. If you rearrange the sequence, then you get a different meaning. And we're going to talk about that much more later on, but that's kind of the general good analogy for how DNA is encoding a message. Okay, so the process of transcription is taking DNA and using it as a template to create an RNA. Now, the RNA specifically that we're going to make from a gene is an mRNA. The M stands for messenger. So that's all going to happen in the nucleus. But the mRNA has to exit the nucleus in order to be read and the message turned into a protein. So the mRNA exits the nucleus through a nuclear pore. The nucleus has little tiny holes in it that allow the mRNAs to escape. And in the cytoplasm, it's going to encounter a ribosome. Now, the ribosome is composed of two subunits, the large subunit and the small subunit. It kind of looks like a little hamburger. And the ribosome is going to do a very important process called translation. Translation is when we take an RNA and we synthesize from it a protein. So the RNA, the mRNA, is also encoding a message, and there are a bunch of free amino acids in the cytoplasm, just beep-bopping around. And remember, there are 20 different types of amino acids, and the mRNA message is going to tell the ribosome which amino acid to pick out of the cytoplasm and attach into a chain. The bonds that form between amino acids, remember, they're called peptide bonds, and when we string them together, we get what's called a polypeptide. A polypeptide is the primary structure of a protein. Now, if this protein is destined for the cytoplasm, the ribosome is going to stay in the cytoplasm and synthesize its polypeptide, and they're going to be what are called chaperone proteins that are going to be associated with the ribosome, and they're going to fold this polypeptide into a functional protein. However, our polypeptide, our catalase enzyme, is destined to live a life outside of the cell. So instead of translating this message in the cytoplasm, the ribosome is going to move to the rough endoplasmic reticulum, the RER. And the ribosome is going to bind to the outside of the rough ER, and as it synthesizes its polypeptide, it's going to push it into what's called the lumen, or the space within the rough endoplasmic reticulum. And it is here in the rough endoplasmic reticulum that the polypeptide will be folded into a protein. But this protein is not fully developed. It is immature. And in order to mature into a functional quaternary structure protein, we have to move from the rough endoplasmic reticulum to the Golgi body. So what happens is a piece of the rough endoplasmic reticulum pinches off, holding this immature protein inside, and becomes a transport vesicle. This transport vesicle is going to bring the immature protein to the Golgi. So you can see here, we have our tiny little ribosome bound to our rough endoplasmic reticulum. Inside of the rough endoplasmic reticulum, you can see there are these little pink dots. They represent our protein. So a piece of the rough endoplasmic reticulum buds off, holding our immature protein, and then the vesicle is going to come and fuse with the Golgi body. Now, the Golgi, you might see this just written as Golgi or Golgi body or Golgi apparatus. It's a series of membrane-bound sacs that are kind of squished flat like pancakes. And the transport vesicle is going to fuse with one of the sacs. And then little sacs will bud off of this and then fuse with the next sac in the line, and then the next sac in the line, and the next sac in the line. And the protein is going to move through the Golgi in this manner, with tiny little vesicles budding off from one section of the Golgi and then fusing with another section of the Golgi. Now, the Golgi is going to be sorting the proteins, so it's going to be aggregating them, collecting them, parsing them out into different places, kind of like a shipping and receiving center, and then it's also going to be modifying the proteins. So if we have an immature protein coming from the rough endoplasmic reticulum, it's going to be maturing in the Golgi body. And this could mean a couple different things. It could mean that the quaternary structure of the protein is being put together. So remember, we have the primary structure, which is the sequence of amino acids. The secondary structure is just one folding motif. So we talked about alpha helices and beta pleated sheets. The tertiary structure is two or more motifs in a put together. And then a quaternary structure protein is two or more subunits that are associated with one another. So in the Golgi, we might have a quaternary structure protein be created. We also might cut a protein in half in order to activate it, because some proteins need to be cut in order to become active. We might add a sugar group. All of this is going to happen in the Golgi, and this is part of the processing. of proteins. Okay, so by the time the protein has moved through the Golgi body, it is mature and it is ready to go off into the wild world of the cell or extracellularly. So these proteins here are going to fuse with another organelle. This is probably a lysosome. So this vesicle is going to be bringing proteins to the lysosome. Maybe they are proteins that are going to be digestion proteins, and the lysosome needs that. But the proteins that we're interested in are in a transport vesicle that is headed to the outside of the cell, the plasma membrane. So here is our catalase protein. Here is the plasma membrane or the cell membrane. And this vesicle is going to fuse with the plasma membrane and release our catalase protein extracellularly. So this is now an extracellular protein. And that is how we go from a message in the DNA to a protein that is outside of the cell. Okay, you guys are going to bring this all together in an image that you look at in class next week. Oh, I guess I should say RER here. This is the RER. Boop. Okay, that's all.